Okay, so welcome back to this next video in which we are discussing voltage-gated potassium channels. Okay, right, so we have just looked at the structure of a voltage-gated potassium channel, and we've seen how voltage-gated potassium channels are built out of tetramers of these alpha subunits of voltage-gated potassium channels. Okay, and this makes up the fundamental structure that you have to have in your voltage-gated potassium channel. Okay, we will come back to the beta subunits in a moment. Okay, so what we firstly want to discuss is these alpha subunits, okay, because the really simple thing would be if there was just one alpha subunit of voltage-gated potassium channels, and that could be called the alpha subunit of the voltage-gated potassium channel. Okay, but it's not that simple, unfortunately. Instead, there are 40 different alpha subunits of voltage-gated potassium channels, okay, which are coded for by separate genes. Okay, now these 40 different alpha subunits of voltage-gated potassium channels are grouped into families to help us try and understand it. Okay, so you have 12 subfamilies, and I'm now going to run you through the 12 different subfamilies and their members. Okay, right, so let me start off with the first subfamily. So firstly, let me just introduce you to the nomenclature. So firstly, you start off by putting a K, okay, for potassium, then you put V for voltage, and then the first subfamily is going to be KV1. Okay, so we're now talking about the KV1 subfamily of alpha subunits of voltage-gated potassium channels. Okay, right, so there are 8 members in this family. Okay, there is KV 1.1, that's the first member, then there will be KV 1.2, okay, and then it will go on all the way up to KV 1.8, so it's quite sensible naming. Okay, that's the first subfamily. Then we'll move on to the second subfamily. The second subfamily is KV2. There are 2 members of this subfamily, and I'm going to stop underlining it and instead just have a colon like so, otherwise I'll run out of space. Now, there are 2 members in this KV2 family, KV2.1 and KV2.2. So that now takes us up to 10 different alpha subunits so far. Only another 30 to go, okay, so let's continue on. KV3 then, okay, there are 4 different members in the KV3 subfamily. So it goes KV 3.1 all the way up to KV 3.4. Okay, then onto the KV4 family. There are 3 different members in the KV4 family. Okay, KV 4.1 all the way up to KV 4.3. Okay, so KV 4.1, KV 4.2 and KV 4.3. Okay, KV5 is the only family which has only 1 member in it. Okay, and this is just called KV 5.1. Okay, KV6 has again 4 members in it. Okay, so KV 6.1 all the way up to KV 6.4. Okay, so let's just count up how many we've got now. So we had the first family that had 8 members in, the second family had 2, that took us up to 10, then we've got 4 from KV3, that takes us up to 14, okay, so far. Then add another 3 onto that, we're up to 17, then 18, add another 4 onto there, 22, so we've only got another 18 now to go, so we're halfway through now. Okay, so then up to KV7 subfamily. KV7 is quite a big subfamily. It contains 5 different members. So KV 7.1 all the way up to KV 7.5. That takes us up to 27 different alpha subunits of voltage-gated potassium channels. KV8 subfamily is a very small one that just contains 2 members, KV 8.1 and KV 8.2. Okay, taking us up to a total of 29 alpha subunits of voltage-gated potassium channels. Then on to the 9th subfamily, KV9. Okay, this contains 3 members, KV 9.1 all the way up to KV 9.3. Okay, that takes us up to 32 different alpha subunits of voltage-gated potassium channels. Then onto the 10th subfamily, KV10, that contains 2 members, Kv10.1, and Kv10.2. Okay, taking us up to a total of 34 alpha subunits of voltage-gated potassium channels. And then finally, both the 11th and the 12th subfamily both contain 3 members to take us up to a total of 40. So Kv11.1 all the way up to Kv11.3, and then the same for Kv12. It starts off with Kv12.1 and then goes all the way up to Kv12.3. Okay, so that's our grand total of 40 different genes coding for 40 different types of alpha subunit of voltage-gated potassium channels. So, all of these different genes will code for alpha subunits which will have a different primary structure. They will have a different sequence of amino acids from one another. Okay, however, they will all fold into a structure that looks like this. They will all have a tetramerization domain, they'll all have the same membrane spanning topology. So these broad structural features that we've discussed here are common for all 40 subunits. Okay? So these 40 subunits can form a quarter of a voltage-gated potassium channel, basically. Okay, so now what we need to discuss is how do you tetramerize them together, because these are the individual subunits. To build a voltage-gated potassium channel, you have to combine 4 of these together. So is it a case of pick and mix? Can you pick whichever ones you like and stick them together? Well, the answer is no. There are general rules for how nature actually constructs voltage-gated potassium channels. The other thing I should have stated is that these are the different alpha subunits of voltage-gated potassium channels in humans. I never explicitly said that anywhere, okay, but I assumed that that was implicitly obvious. If you go into other species, the voltage-gated potassium channels will be different, okay, they'll be called different things, you'll have fewer potentially. Okay, this is what you have in humans. Okay, right, so let's now discuss then how they tetramerize together. So the first thing that I want to discuss is the simplest case. Okay, so the simplest case is that you just take 4 identical subunits, okay, and tetramerize them together. Okay, and this forms you what is known as a homotetramer. So homo means same, tetramer means 4-membered structure. So this is a tetramer, a voltage-gated potassium channel tetramer, where the 4 subunits that comprise it are all an identical one, basically. You pick one and you use it 4 times, okay? And that builds you a homotetramer. Okay, so most of these alpha subunits that I have just listed off here can actually form homotetramers, okay, and those will be functional voltage-gated potassium channels. So what I'm going to write out is the exceptions, okay, which ones cannot actually form homotetramers. Okay, so there are certain families that cannot actually form homotetramers, okay, so they are the Kv5 family, the Kv6 family, the Kv8 family, and the Kv9 family. So if I underline these, here is 5, here's 6, here's 8, and here's 9. The members of these 4 families, this family, this family, this family, and this family, cannot form homotetramers. So what that means is if you, for instance, take the gene for Kv5.1, and you put it into some cell that has no alpha subunits for voltage-gated potassium channels in it, okay? So we're talking about genetic engineering experiments here. You are in some cell culture laboratory, you have cells growing in some incubator, okay, and they have no genes for voltage-gated potassium channels because you have manipulated it so. And you put in this gene for Kv5, okay? You do not find the cell having voltage-gated potassium channel tetramers, basically. Okay, if this was capable of forming homotetramers, you would expect that when you put in the gene for Kv5.1, then you would end up with Kv5.1 homotetramers, i.e. voltage-gated potassium channels where there were 5 of these constructed together. You do not find that. Okay, that's what I mean by these do not homotetramerize. That when you do this experiment using genetically engineered cells, you don't find functional voltage-gated potassium channels produced for any of the genes in these 4 families. Whereas, if you, for instance, took Kv 1.1 and did that, put it into these cells that have had voltage-gated potassium channel genes all removed, then you would find functional voltage-gated potassium channels being produced that were homotetramers of Kv 1.1. Okay, right, so those are the 4 families which don't actually form homotetramers. Okay, now what I'm going to talk about is the more complicated situation. Okay, which is that you can also form heterotetramers, okay, which are voltage-gated potassium channel tetramers where the 4 subunits are different. Hetero means different, okay, so they're not all the same identical type of subunit anymore. Okay, so certain families can form heterotetramers within the family. Okay, so let me firstly just write out the families that participate in these heterotetramers, okay, within families, and then I'll explain in more detail what it means. So Kv 1, 7, and 10 are the families which are capable of forming intra-family heterotetramers. So Kv 1, Kv 7, and Kv 10 here, the ones underlined in green. Okay, so these families are capable of forming heterotetramers. Okay, so voltage-gated potassium channels where the 4 subunits are not identical. Okay, you've used different types of subunits in the different slots. Okay, it doesn't have to be the case that you use 4 completely different subunits, okay? It just means that they're not all the same. Okay, so for instance, you could use 2 Kv 1.1s in this and 2 Kv 1.2s. Okay, that would be a heterotetramer because it wouldn't be a homotetramer. Okay, it doesn't mean that I've picked one to go here, a different one to go here, a different one to go here, and a different one to go here necessarily. Okay, so you can form heterotetramers using subunits that are in the same family. Okay, so I can build a voltage-gated potassium channel where these 4 subunits are from the Kv7 family. Okay, or from the Kv1 family, or from the Kv10 family. What you don't find is mixes. You don't find voltage-gated potassium channels which have some Kv1.1s in, and then some Kv7.1s in. Okay, there's no inter-family mixing. It's intra, within the family, heterotetramers. Okay, so you can build heterotetramers which are intra-family heterotetramers. Okay, so where the different subunits are all from a single family, basically. They're not all identical, but they are at least in the same family. Okay, right. Now what I'm going to talk about is inter-family heterotetramers, okay, which you can form. Okay, so the good example of inter-family heterotetramers involves these families that I have talked about up here, the ones that can't form homotetramers. So I'll bring them back down here. Kv5, 6, 8, and 9 subfamilies. These have a special name, okay? These are known as modifier alpha subunits, okay, because they can't form homotetramers on their own. Okay, they can only form heterotetramers with subunits from another family. Now, which other family do they need? Well, they are all reliant on members of the Kv2 family. Okay, so this family here. So basically, to use these modifier alpha subunits, i.e. the members of these 4 families, Kv5, Kv6, Kv8, and Kv9, okay, to actually use them, what you have to do is you have to build a heterotetramer where you have some subunits from these families, and then some subunits from the Kv2 families, particularly Kv2.1. Okay, so Kv2.1 is the one which can build voltage-gated potassium channels where you have some of these modifier subunits in. Okay, so these have to form heterotetramers, basically. They can't form homotetramers. Right, okay, so that's the alpha subunits now discussed, and we've discussed how you can actually tetramerize them to form a voltage-gated potassium channel tetramer. What I now want to talk about is the beta subunits. Now, the beta subunits are what are known as accessory or auxiliary subunits. Okay, they are not absolutely necessary to have in order to build a functional voltage-gated potassium channel. In order to build a functional voltage-gated potassium channel, all you need to do is tetramerize 4 alpha subunits together. However, adding on beta subunits can modify the properties of voltage-gated potassium channel alpha subunit tetramers. Okay, they can change the threshold potential for the activation of the voltage-gated potassium channel, for instance. Okay, so the accessory subunit that I am going to talk about, and it's worth saying that there are other accessory subunits for voltage-gated potassium channels, but the accessory subunit that I am going to talk about is the beta subunits. Okay, so I'm only going to talk about one, so maybe I should cross off the S. We're going to talk about beta subunits. Okay, and there are other classes of accessory subunits. Okay, but it's the beta subunits we're going to talk about. Now, there are 4 different beta subunits for voltage-gated potassium channels, and these are known as KV betas. So you put KV, voltage-gated potassium channels, then you put a beta after the V to emphasize the fact that we're no longer talking about alpha subunits, but beta subunits. And the 4 different ones are called KV beta 1.1, KV beta 1.2, and then you also have KV beta 2 and KV beta 3. Okay, so those are the 3 different types of beta subunits of voltage-gated potassium channels. And as I say, adding these on to a voltage-gated potassium channel alpha subunit tetramer can modify the properties of that alpha subunit tetramer. So let me show you where these are added on. Basically, they are going to be added on to the tetramerization domains. Okay, so what you can do is you can stick a beta subunit, and I'll put it on like this. So you can stick a beta subunit underneath your tetramerization domain here. So here is one beta subunit attached to this red alpha subunit's tetramerization domain here. So this, in vivid purple here, this is a single beta subunit. And of course, every single one of these tetramerization domains can end up with a beta subunit attached to it. So now let's draw one in for this blue subunit here. Okay, so here it is. Here's the blue subunit, its beta subunit. Okay, and then the turquoise subunit round the back there will also end up with one, and of course the orange one as well. Okay, so you'll end up with 4 separate beta subunits attached onto the alpha subunit tetramer here. Okay, and they're all shown in vivid purple. And that then produces our final voltage-gated potassium channel complex, where we have 4 alpha subunits complex with 4 beta subunits. Again, this is the full voltage-gated potassium channel, although, as I say, you can add on more accessory subunits. But I want to emphasize once again that you don't need the beta subunits to form a functional voltage-gated potassium channel. To form a functional voltage-gated potassium channel, you only need the tetramer of alpha subunits. That is essential. You can't get away with 3 alpha subunits. You have to have 4 to form the full channel. Okay, right. So that now is enough on the structure of the voltage-gated potassium channel. What we now want to turn our attention to is the opening mechanism. Okay, so why does a depolarization of the electrical potential difference across the cell membrane cause the voltage-gated potassium channel to open? Okay, so I'll get a new piece of paper here. Right, so firstly what I'm going to do is I'm going to draw a picture that I haven't drawn before, which is, I'm going to draw a very basic picture which will represent what you would see if you were to cut down the middle of the voltage-gated potassium channel. So I'll just get it back for a second. Okay, so what we're effectively going to do, and I'll use this picture here, what we're effectively going to do is we're going to put our carving knife, if you can imagine, one end of it will be here, the other end of it will be like this, and we're going to chop right down through the middle, and I'm going to have a look at the exposed surface. Okay, I'm going to have a look at that view of the voltage-gated potassium channel, which is something that I haven't drawn previously. Okay, now we'll start by drawing it in the open state. Okay, and then I'll show you how the closed state will be different and why this isn't going to allow potassium ions to move through. And then we'll talk about how you move from being in the closed state to being in the open state when the electrical potential difference across the cell membrane is depolarized. Okay, so let's start off with what you would see if the channel was in the open state. Okay, so remember we're taking this new view where we've chopped down the middle. Okay, like so. And I'm just drawing the pore portion. I'm not interested in the voltage sensing domains. Okay, so this is just the pore portion. Right, so this is the extracellular side, and I'll put the membrane here. Okay, this is the membrane. Okay, so effectively, if I just completely make it clear what I have done here, we're using this very simple picture where we're just showing the pore portions. I'm not interested now in the tetramerization domains, I'm not interested in the voltage sensing domains, I'm certainly not interested in the beta subunits. All I'm interested in is the pore forming domains, and I've effectively chopped down the middle of that, and I'm having a look at what we're seeing. Okay, so this is the extracellular fluid side. Okay, and this is the cytoplasmic side. Right, so you can see that on the extracellular fluid side of the voltage-gated potassium channel, the pore is very narrow. Okay, now this portion of the pore, which is very narrow, has a special name. Okay, this is known as the selectivity filter. And as its name suggests, it is very important for the selectivity of voltage-gated potassium channels. It is important for why voltage-gated potassium channels are selective for allowing potassium through rather than other cations. Okay, and we will come back to the selectivity filter later on. So it's the narrowest portion of the pore. For now, let's ask ourselves the question, why is it narrower than the lower down portions? Well, remember the structure of the voltage-gated potassium channel. Let me bring this picture back again. What was the portion that was closest in? It was this P-loop. The P-loop did not go all the way through the membrane. It turned back on itself. So this innermost portion, consisting of P-loops here, so there's another one here and another one here, this is only on the extracellular side. Okay, once you get further enough through, okay, you get past the P-loop here, and now what's lining the pore? Well, it's the S6 helix that's behind. Okay, so if we colour this in, if we transfer this information onto this picture, here is the P-loop in pink here, like so, on both sides. Okay, then sitting behind the P-loop, you've then got the S6 helix here in blue. Okay, and of course you'll have 4 of these, one here and one here. Okay, and then behind the S4 helix, you've then got the S5 helix in green here. So on the extracellular side of the voltage-gated potassium channel, it's the P-loop which is lining the pore. Okay, and this is why the pore is narrowest here. And then on the intracellular side, it's S6 that's lining the pore, and this is the thicker portion of the pore here. Okay, right, so I'll get rid of that now. So, this is the channel in the open state. Okay, so this is the open state channel. Potassium ions can move through this. Yes, they've got to get through this very narrow selectivity filter portion, but this is an open channel. So what does the closed state channel now look like? Okay, so let's now draw the closed state. So, what changes between the open state and the closed state is the S6 helices. The S6 helices are going to move inwards in the closed state to obstruct this intracellular aspect of the pore. Okay, so let me show this nice and simply. So once again, here is the P-loop portion, the selectivity filter, and now I'll show this like so. Here is the S6 helix moving in and obstructing the intracellular portion of the pore here. Okay, like so. So let's again colour code this. So here, the portion making the narrow selectivity filter here, this is the P-loop in vivid purple here. Okay, now we have our S6 helix coming in closer here, okay, and obstructing the intracellular aspect of the pore here. So all of this in blue here, this is the S6 helix, which is resulting in that intracellular aspect of the pore being closed, and then behind it, not really involved, we've then got the S5 helix, okay, on the peripheral aspect. Okay, so this is the closed state channel. Okay, so what we now want to discuss is how you go from being in the closed state to being in the open state in response to changes in the electrical potential difference across the cell membrane. Okay, well, as the name suggests, the voltage sensing domain, the S1 to S4 portion of the voltage-gated potassium channel alpha subunit, this portion is responsible for sensing the change in electrical potential difference, and it's then going to convey this change to the pore-forming domain via the S4-S5 linker. Okay, so basically this portion is going to change in conformation, or rather all four of these portions are going to change in conformation, and they're then going to transmit changes in conformation to the pore-forming domains, which will cause the S6 helix to move outwards away from the pore, and then they'll stop obstructing the pore, and then you'll move from the from the closed state to the open state. And this movement from the closed state to the open state is called activation. Okay, right. So, let's discuss this in a little bit more detail. What is the actual change that is going to occur in the voltage sensing domain? Well, basically, the voltage sensing domain is going to move outward. Okay, so if I start actually showing a voltage sensing domain here. Okay, so here are our voltage sensing domains. So remember, they sit peripheral to the pore forming domain. So I'm just showing two of them here. Okay, which are the two we'll see if we chop through. Let's say we've chopped through at an odd angle like this. Okay, and we're seeing one voltage sensing domain here and one here. Okay, so the voltage sensing domains contain an important alpha helix, which I'll show in orange. Okay, which is the S4 alpha helix, and it's this one that is going to respond to the depolarization in the electrical potential difference across the cell membrane. So let's say in orange here, this represents S4. Now S4 has some important properties. S4, in absolutely all alpha subunits of voltage-gated potassium channels, has 4 positively charged amino acid residues within it. Okay, now these positively charged amino acid residues are either arginines, okay, or lysines, okay, which are both positively charged amino acid residues. So you're going to have 4 of these. Okay, so I'll put these on 1, 2, 3, 4. Okay, and on this one as well, 1, 2, 3, 4. Okay, so let me now just show you the structure of arginine and lysine, just to make sure that you're familiar with those and understand that they are positively charged amino acid residues. Okay, so I will start with arginine, the more complicated one, and I will show it as a residue, okay, which means that I'm going to show it as though it is within a polypeptide. Okay, I'm not just going to show a free arginine amino acid. Okay, I'm going to actually show the amino acid as though it's within a polypeptide. Okay, so here is the amino group here, which will be bound to the carboxylic acid group of the amino acid prior to it. Okay, then after the amino group, you'll have the alpha carbon with a hydrogen coming off it. Then after the alpha carbon, you'll then have the carboxylic acid group, which will be bound to the amino group of the next amino acid along. Okay, so that's what I mean by showing it as a residue, that I've shown these groups as though they're bound to other amino acids by peptide links. Okay, so what's the R group then of arginine? This is the important bit. Okay, arginine firstly has 3 methylene groups. Now, because I don't want to have to draw out 3 methylene groups, I'm going to draw 1 methylene group, bracket it like so, and then subscript it 3. That's a useful trick. It just means repeat this methylene group 3 times. Okay, and then after that, what you're going to have is a nitrogen atom with a hydrogen atom coming off it, and then following that a carbon atom, which will have an amino group coming off it, like so, and then a double bond also to a nitrogen atom, which then has a hydrogen coming off it. Now, so far, we have not got a positive charge there. However, what you need to remember is this nitrogen here that is double bound to that carbon. This is going to have a lone pair of electrons on it. Okay, now, before I just talk about this, let me just say that this nitrogen has a special name. Okay, this is what's known as the guanidino-nitrogen. Okay, the nitrogen that is double bound to the carbon there in the arginine structure. So that's the guanidino-nitrogen. Okay, now the guanidino-nitrogen here, okay, has a lone pair of electrons on it, which I'll show here. Okay, and this lone pair of electrons is a center of negative charge. Okay, so what can end up happening is free protons in solution can end up coming and associating with this lone pair of electrons, okay, because it's a lovely center of negative charge, and it's a lure for that positively charged proton here. Okay, and when you end up with this proton attaching onto the arginine head like so, that ends up giving the whole arginine a positive charge. Okay, because previously the arginine was neutral, and now a proton has come and associated with a lone pair, and that makes the whole thing positively charged now. Okay, it's a similar story for lysine. So this is the amino acid residue arginine here. Okay, let's now show lysine. So again, I'll draw out the core amino acid structure firstly. So here's the amino group. Here's the alpha carbon with a hydrogen coming off it, and I'll just make sure this is at the right height. Here then is the carboxylic acid group, which will be attached to the amino group of the next amino acid along. And then the R group of lysine consists of 4 methylene groups firstly. And again, I'll use my trick of bracketing that and putting a 4 here. And then you'll have a amino group here. Okay, and again, this nitrogen of this amino group will have a lone pair of electrons which a proton can come and associate with, giving the head of the lysine a positive charge. Okay, so that's how lysine and arginine residues end up getting a positive charge. Okay, so in these S4 helices within the alpha subunits of voltage-gated potassium channels, you always have 4 arginines or lysines, 4 positively charged amino acid residues. And you might be asking, well, why can't you tell me exactly which amino acids they are? Okay, remember, we are talking broadly about 40 different types of alpha subunit here. Okay, so in different alpha subunits, which amino acids they specifically are will vary. Okay, but the running theme is that all of them in their S4 helices have 4 positively charged amino acids, either arginine or lysine there. Okay, which gives the S4 alpha helix a positive charge. Now, what then happens when the electrical potential difference across the cell membrane depolarizes? Okay, so let's say the electrical potential difference across the cell membrane has now depolarized. Okay, well, what does that mean? Let's remember what that means. That means that how negative the electrical potential difference across the cell membrane is has gone down. Okay, so originally let's say it was around negative 65 millivolts, and let's say it's now gone up to negative 40 millivolts. Okay, that means that the electrical potential intracellularly is now only 40 millivolts lower than the electrical potential extracellularly. Okay, so how much lower the electrical potential intracellularly is compared to the electrical potential extracellularly has gone down. Okay, now what does this mean for the S4 helix? Well, the S4 helix has 4 positive charges on it. Okay, that means that the S4 helix is going to be attracted to the intracellular compartment because the intracellular compartment has a lower electrical potential difference. Sorry, not a lower electrical potential difference, a lower electrical potential. There's an important difference between those two concepts. A lower electrical potential than the extracellular compartment. Okay, so S4 is continually going to be feeling this attraction downwards, and it's going to be sort of pulled down into the cytoplasm. Okay, it's like a spring that's sort of being pulled downwards, basically. Okay, and then when you reduce how negative the electrical potential difference across the cell membrane is, when you depolarize like this, that's going to suddenly mean that the attraction on the S4 helix, that's attracting it into the intracellular compartment is going to be hugely reduced, because now the intracellular compartment's electrical potential is only 40 millivolts lower than the extracellular compartments. Okay, so the force on the S4 helix that's driving it down has been reduced, basically. And now what's going to happen is it's going to recoil like a spring, basically. You're releasing the spring a little bit and it's going to recoil back. So the S4 helices move up. The entire voltage sensing domains move up, basically. And this movement up is then going to be transferred to the pore forming domains here, okay, via the S4, S5 helix. Okay, so remember there is this linker between the S4 alpha helix and the S5 alpha helix here. So when all these voltage sensing domains move up, what's going to happen is the S4, S5 linker here is going to also be moved upwards, and that's going to cause a conformational change in the pore forming domain, and that somehow then leads to the S6 helix being pulled outwards, okay, and then it goes from being in the closed state to being in the open state, and therefore it is now activated. Okay, right, so just a little bit more nomenclature then, okay. When the electrical potential difference across the cell membrane is returned back down to negative 65 millivolts, okay, what will then happen is the S4 helix will be pulled back down, because now it's going to feel a greater attraction once again back into the intracellular compartment, okay, and then the reverse conformational change will occur, and you'll go back from being in the open state to being in the closed state, where the S6 helix is obscuring the intracellular portion of the pore. That process of returning back to the closed state from the open state is not called inactivation. Instead, it is called deactivation. Okay? So do not confuse deactivation with inactivation. Deactivation is the closing of the channel because the voltage stimulus that was causing it to open has been removed. Inactivation is a totally different process, and we will come on to inactivation later on. Okay, so activation and deactivation are two important pieces of nomenclature. Okay, the other thing that I want to talk about is that this is what's known as the V-gate. Okay, you can't read a paper on voltage-gated potassium channels without hearing someone referring to the V-gate. Okay, this way of closing the channel by moving the S6 helices inwards and stopping potassium moving through in that way, that's referred to as the V-gate of the voltage-gated potassium channel. The V refers to the fact that it's gated by voltage. Okay, there are other gates, such as the one that's responsible for closing the voltage-gated potassium channel in the process of inactivation, and that's called something different. It's called the N-gate. Okay, we're going to look at N-type inactivation. There are even other types of inactivation beyond that, so there are more gates. Okay, we're not going to talk about those because they're not very well understood. Okay, but that's just another piece of nomenclature. So if you hear people referring to the V-gate, it literally is just referring to this mechanism of the S6 helices closing the voltage-gated potassium channel when the electrical potential difference across the cell membrane is hyperpolarized of negative 40 millivolts, i.e. more negative than negative 40 millivolts below the threshold potential. Okay, right. So we'll call it there for this video, and in the next video what we'll do is firstly discuss the permeation mechanism and the origin of the selectivity for potassium ions, and then what we'll do is discuss N-type inactivation.